Okay, thank you very much. So I'm a postdoc in the College of Science, working in biology with Carlos Garcia de Lenes and Sonia Consuegra. And I'm going to talk to you today a little bit about my research, which has been using next-generation sequencing technology to basically to look at how we can understand how stress affects fish health and how we can potentially improve that in terms of aquaculture and fish farming. So first of all, I'm sure most of you have heard of next-generation sequencing. So, I mean, this has been a real revolution for biology. You know, in the last 10 years or so, it's revolutionized many aspects of biological research. And basically what we mean by next-generation sequencing is DNA sequencing, but on a very high throughput scale. So before the advent of this technology, you know, we could sequence DNA, but it was on a very targeted basis, you know, basically single molecules at a time. So when we think about, for example, when the human genome was first sequenced, it was sequenced in targeted bits at a time and gradually put together over a long time, and it took, you know, a lot of effort doing it that way. But now with this new technology, we can sequence a human genome or hundreds of different bacterial genomes all in one go with one run of this machine. So what we get out of it is relatively short reads, so they're only sort of a couple of hundred base pairs long, but we get, for every part of the genome, we'll get coverage of thousands, hundreds of thousands of times for each one. So it's like a jigsaw. We get all of these millions of reads coming out of it, and we want to put them all back together again. So you can see we can get hundreds of millions of sequence reads from one run of the data, which can be up to, you know, terabytes worth of data. So that is basically why we need high performance computing. It's because it's such a vast volume of data. In order to store it and to process it, we need massive computer power and massive memory. So in terms of all the different applications of next generation sequencing, there's lots of different things you can do with it. So obviously you've got genome sequencing, or resequencing, and this is used for gene discovery, sort of working out what genes do and sequencing genomes for new animals and plants. It's used a lot in evolutionary biology, so we can see how closely the genomes of different species are related to each other, and also within populations of the same species, you know, how closely are populations related to each other. And then we've got RNA sequencing. So RNA is like the active part of the genome, so it's the transcribed part of the genome. So we can use RNA sequencing to look at global gene expression, which gives us an idea of what the cells are actually doing. Another type of sequencing is methylation sequencing. So DNA can be packaged in different ways, and one of the ways that it's modified is addition of methyl groups actually onto certain bases of the DNA. And this affects how the DNA is available to be transcribed. So if we sequence DNA with methylation sequencing, we can see how it's likely to be active and expressed. We can also look at DNA and RNA protein interactions. So for example, this can be used to see how transcription factors bind to receptors. And again, proteins called histones are really important in packaging up the DNA, which makes it available to be expressed or not. So this is another form of epigenetics. And then finally, we've got metagenomics. So this is a way of analysing what is present in complex communities. So for example, I work a lot on the microbiome. So you've got lots of different bacteria present in your gut. If you sequence the DNA from everything, you can work out who is present in that complex community. And another example that we do in our lab is called eDNA metabarcoding. So this is environmental DNA. If we take just a water sample from the environment, we can extract the DNA from it, we can match it against databases and see what animals and plants and invertebrates are all present in that water sample. So this is really useful for ecology as well. In terms of my research, as I mentioned briefly, I'm basically trying to improve, see if we can understand how stress affects fish health and how we can improve that. In terms of aquaculture, probably the major challenge of aquaculture is disease. Disease is a really massive problem in terms of fish welfare and health, in terms of releasing pathogens into wild populations potentially, and also obviously the economic cost of loss of fish to disease. So in order for aquaculture to be more sustainable in the future, it's a real priority for us to try and improve disease resistance. So what I've specifically been doing in my research is looking at different molecular mechanisms by which stress affects phenotype. So phenotype is, you know, your overall health and disease resistance, but there's lots of different levels of biological organisation below that. You go from the genome through your epigenome, your transcriptome. I haven't mentioned the proteome. But if we look at these different levels, we can look at the different mechanisms which might be contributing to the overall effects on fish health. And also another one is the microbiome. So this is the host-associated microbial communities. And importantly, all of these three things I've mentioned, the epigenome, the transcriptome, and the microbiome, we know are very strongly affected by the environment. So if environmental factors, if stress is affecting these different things, it might eventually change health and disease resistance. So I'm going to talk mainly about the microbiome. That's what most of my research is on. And I just thought we'd just start with a very brief definition. So the microbiome is basically all of the microbes which are present in or on the human body or another environment. And they include commensal organisms, symbiotic, and pathogenic organisms. And it's pretty amazing, really, when you think about the number of bacteria and other things we've got associated with us. There's about 10 times as many bacterial cells on the human body as there are human cells, which is incredible. And in terms of the number of genes that they contribute, there's about 100 times more bacterial genes on the human body as there are human genes. So that just shows the massive potential that microbiome has on human health. And that's just considering bacteria. You know, there's also eukaryotes and archaea and viruses, but we know very little about them, really. So, yeah. So, I mean, as I said, there's, it's been increasingly recognised that the microbiome is absolutely vital for host health. For example, it can enhance digestion, nutrient uptake, and it's really important in pathogen defence as well, and for the immune system. But also that relationship between the host and its microbiome is really sensitive. It's a really delicate, finely balanced relationship. So it could be really sensitive to environmental stresses, and potentially, if it's disrupted, that might cause adverse health effects. And you might have heard of the holobiontic concept, which is basically thinking about the host and its microbiome as one combined organism. And I think this is going to be, you know, a really important thing for future medical research, really. Considering, you know, when you treat a patient, you treat its microbiome as well. So considering that whole thing as one. So mainly due to next-generation sequencing, there's been a real explosion in microbiome research over the last few years. So far, it's mainly based on a small amplicon of the 16S gene, so it's only very targeted regions that we're sequencing, but that's enough for us to tell basically who is there. In the future, it's going to be more about what are they actually doing. So in order to do that, we can look at bacterial gene expression, which is the metatranscriptomics, as well as specific genomics and metabolomics as well, probably. And it's really quite cool. There's lots of really big consortium projects which are contributing a lot to microbiome research. So, for example, in America, there's this project called American Gut, where if you want to, you can send off your own sample of your own microbiome, get it sequenced for $100, and then they'll tell you what's in it and how it compares to the rest of the population. But all this information is being collated from lots and lots of different people, which is building up massive databases for the human microbiome. So we can see, you know, how different it is on different parts of the body, and also with development and age as well. And you know, we know that the microbiome is very dynamic. It's very sensitive to lots of different things. We know it changes with diet, with lifestyle, everything you can think of, really. It seems the microbiome is involved in it. And these are just a few sort of headlines I picked out. You know, there's been a lot of media interest as well, and it seems that the microbiome has kind of been linked to everything, really. You know, potentially bacteria can make you smarter. It's linked with heart disease, changes your mood, it's strongly linked with obesity. So there's all these really interesting avenues of research. And if you're interested, I really recommend this book. It's a really, really good book on the microbiome. So what about the fish microbiome? So obviously there's been a lot less research done on the fish microbiome, but we know that the microbiome associated with the intestine, but also the skin and the gills are really important for fish health. We know it varies with various host-specific factors, so things like age, with genetics, and the immune system, but we also know that it changes a lot in response to different environmental conditions. So it changes with season, with diet, and with stress as well. And in turn, this can then affect important things like nutrient uptake and metabolism and growth, which are all really important for fitness. And potentially as well, thinking about the immune system and disease resistance, which is what I'm interested in. So when we think about how we actually analyse the microbiome, so the data which comes out of the sequencer looks a bit like this. So this is a FASTQ file, which is essentially it's an actual the sequence plus quality information. So this is 3 sequences, I think, but what we actually get back is can be 20 to 30 million sequences per individual, times lots of replicates, times lots of treatments. So you're looking at 500 million sequence reads easily to deal with. And the first step that we perform, so this is all done with sort of open source bioinformatic tools. First step, we do some quality control, filtering out the bad reads and contaminants and things like that. And then we align all of the different reads to existing bacterial databases. So all those existing genomes which are out there, we align our reads to them to work out what our reads are. And then we cluster them. So we use these things called operational taxonomic units, which basically it's usually done at 97% sequence similarity. So you say if one sequence is 97% similar to something else, then it's the same thing. So we cluster into these operational taxonomic units and we annotate them. So we assign them to a genus usually. And then downstream of that, you can perform sort of ecology measures really. So we're looking at the number of bacteria which are present and the diversity of the communities, and also overall how the whole structure of the microbiome is and what, a little bit about what that community is doing. So moving on to some of my results. So the first thing I did was I was looking at the microbiome in hatchery-reared fish and wild fish. So I collected populations, 7 different populations from different rivers in the UK and also different hatcheries, and then sequenced the microbiome of all these individuals. And what you can see is the results on the right. So on the top graph, we've got richness, so that's basically a measure of the number of bacteria present in those communities, and the bottom cluster diagram is overall structure of the communities. So they're colour-coded by population, and the bottom graph, it's 1 dot per individual fish. And you can see that within populations there's a lot of variation, so in both terms of richness and in terms of structure. All the different coloured points in the structure are, they're clustering together. So all these populations are quite different from one another. But more strikingly, between the hatchery populations and the wild populations, there was a really clear difference, both in terms of diversity and in terms of structure. So all the reds and the pinks and the oranges are kind of clustering together at the bottom as well. So, and that was quite interesting for us. And we think that probably, it's mainly related to diet. So fish in hatcheries are fed pellet diet, it's an artificial diet, it's the same all the time, whereas in the wild, they get a much more diverse diet. This is interesting because generally, having a more diverse microbiome is thought to be more healthy. So that already puts our hatchery fish at a disadvantage, really. They seem to have less diverse microbiomes. The next thing we wanted to do was see how plastic the microbiome was. So if you change the conditions, does the microbiome change? So what we did was we took some wild fish and we took some hatchery fish and we put them together in 3 different treatments. We had purely hatchery group, a group which was fed with an enriched diet, so it was a more diverse diet, and a group which was actually put in wild conditions. And we could see, so on the left-hand side, again, you've got the richness, so the number of bacteria, and on the right, we've got the structure of the communities. And it was really interesting, particularly in the richness. So these are the hatchery origin fish on the left-hand side and the wild origin fish. But you can see there was no effect of origin at all. It was completely down to what treatment they were fed. So if you enrich the diet, you get a more rich microbiome. With the structure on the right-hand side, again, you can see the 3 different colours are clustering quite strongly apart. So again, it's predominantly down to the treatment, the type of diet they're fed. But we did manage to detect a significant effect of origin as well. So that's probably due to, bacteria that were present in their gut before. So it shows, although they're very plastic, they do retain these signatures of origin as well. So then we were thinking about, we know that they're different between the hatchery and the wild, but what about specific stress that we see in the hatchery environment? So exposed salmon during really early life stages to an acute stress, which was very short term, and a longer chronic stress. So this is the results on the skin microbiome. And the main thing you can see is that on the acute stress on the right hand side, there's a lot more of this bright red bar. So all of these, sorry, I should have said, these are the top 20 OTUs that are present in the community. They're all colour-coded by different bacteria, and each individual bar is a fish. But you can see in the acute stress there was much more of this red bacteria. So these are a genus called Kinetobacter, and also there's one called Eromonas, which is in a slightly pinker colour as well. And both of these genuses increased significantly following that early stress. And what's really interesting is that both of these genuses include many opportunistic fish pathogens, which could be really damaging for their health potentially. And we saw this similar sort of thing in the gut as well. So we saw an increase in these Echinotobacter and a reduction in lactobacillus. So lactobacillus are kind of the good bacteria, and that's what you'll find in probiotic drinks. So it's we found a reduction in the supposedly good ones and an increase in the bad ones, which could be, detrimental for health. Oop, wrong way. This is another interesting thing we found, specifically looking at cortisol, which is the host's stress response hormone. So when fish are stressed, they tend to produce cortisol, which will facilitate their sort of flight or fight reaction, but long-term cortisol production can be damaging as well. And we found that cortisol had a specific effect on the microbiome. So all of these fish have been ordered based on the level of cortisol in their feces along the bottom. So the ones on the right-hand side have got much more cortisol in their feces. And you can see there was a really, really pronounced effect on their microbiome as well. So all of the yellow bars, which were present in low cortisol fish, sort of declined. And those yellow bars are those lactobacillus. So those are those good bacteria. So cortisol is specifically reducing the amount of good bacteria in the gut. And then finally, we were looking at how the microbiome might actually be related to disease. So this is in tilapia, and tilapia are stressed at low densities. They don't like being at low densities. There's more aggression. And we find that this social stress increases their susceptibility to a particular pathogen. And what we also found was a difference in their microbiome as well. So all of the green spots are control fish, so they're not infected, and the red spots are fish which are infected. And so infection with saprolegnia causes a remarkable change in their microbiome, but we also found differences within those densities as well. So it could be that stress related to that density is affecting their susceptibility to infection. So this is something we're currently looking at. So I was just, hopefully I've got time to quickly talk about the epigenome and the transcriptome as well. as I said before, the epigenome, the simple way of looking at the epigenome is just a way of packaging DNA. So we've all got one genome, which is by and large present in all of our cells, but all of our cells do different things. So that's because each cell has a different epigenome. So it's the way that the genome is packaged and the way that it's available to be transcribed, basically. And what's really important is this is affected by the environment really readily. Oops. So we can perform DNA methylation sequencing, whereby we look at where those methylation groups are on the DNA, which gives us an idea of how that DNA is accessible or not. And this is done by a really simple chemical conversion. So we convert non-methylated cytosines to tyrosines, and then sequence them, and then we can tell which are present or not. And in terms of the RNA sequencing, as you know, DNA is transcribed to RNA, and then RNA goes on to produce proteins via translation. But if we capture all of those RNAs in the cell and sequence them directly, we get a measure of like global gene activity, so what the cell is actually doing. So we've been combining these two methods. Haven't really got time to go through it, but essentially it's a similar process as I described last time. We start by, QCing the reads, mapping the reads to the genome, and quantifying the methylation level and the expression. And this is what we found. So this is the same study as we performed with the chronic stress and the acute stress, if you remember the microbiome. And we looked at the epigenome of these fish. And the graph on the left is just an overall level of methylation in the genome. And you can see there's quite differences in the groups. And specifically, if we look at the individual bases that were methylated, we found that chronic stress caused differences one way, and acute stress causes differences another way, including quite a lot of immune genes. So this suggests that they've got differential potential for expression. And in terms of the actual RNA expression, what we did was, after the stress exposure, we then exposed them to a pathogen to test how their immunocompetence, basically, how they responded to a different pathogen. And what we found was that, really interesting, the acute stress actually stimulated their ability to respond to the pathogen. So those immune genes responded quicker and to a greater extent than the control fish, which could be useful for management, trying to improve their immune system, whereas the chronic stress reduced that. And that's it, I promise. Just one more slide. So overall, just using different next generation sequencing technologies, it's really powerful. We can look at all different levels and to look at the different mechanisms of stress and how that might affect disease and health. And accessibility to high performance computing really makes that possible alongside open source bioinformatics pathways. I didn't mention the proteomics and the methylobiomes, but they are there, so it'd be good to include them as well. And in the future, I think really expanding the systems biology approach, the collaboration between biologists with computer scientists and mathematicians to work out the best way of integrating these data sets. So I think that's where the future is. So I'd just like to thank everyone who makes this possible. Thank you.